Sorry, go ahead.
Oh, I know this.
Okay. There's no introduction today. There was not much of it yesterday. Just wanted to say that we have the same procedure that we had yesterday. Sydney talks, and whenever he needs a short break, we'll take a break, and then we have questions and answers.
Yeah.
Until he's tired, and then we'll have coffee and tea afterwards. So I hope you enjoy yourselves as much as yesterday. And Sydney, it's all yours.
Well, today I will discuss how we went from classical molecular biology, if indeed there is such a thing as classical molecular biology, into the studies of the rest of the biology. I'd always been interested in the nervous system, and so once we had finished protein synthesis and all that other stuff, I decided that I would want to stop doing molecular biology and get into doing something else. And so I started to read widely in order to find a suitable experimental organism in which we could go and do the sort of things we'd been accustomed to doing with bacteria and their viruses in order to have an experimental basis for it. And so I read very widely. I looked at all kinds of protozoa, and then I found a class of animals called the micrometazoa. They were all microscopic organisms, and there were two that particularly caught my attention. One were the little nematode worms, and the other were the rotifers. Some work had been done on rotifers, but it looked as though to do genetics on them would be very, very difficult. They had long life cycles. We wanted something that wouldn't take months of waiting till you got the answer. And eventually, after a long amount of reading, I set on the nematode worms. Now, it actually turned out that a rather famous scientist called Richard Goldschmidt had studied a parasite of humans called Ascaris roundworm, and that he'd actually described their nervous system in 1918. And I hurried off to the library in Cambridge to read this. It's printed in something called the Jahrbuch der Zoologie, and he did this as a student of 18 working in a museum in his hometown in Germany. Goldschmidt became rather famous later on for his work in genetics. I was amazed because in those days they never cut the pages of journals. You had to go with a paper, a knife, and really cut the pages. They just printed them, bound them, and sent out the journal. And I found in the basement of the zoology lab this journal, and I found the pages were uncut. So that means they had sat there for over almost 50 years, 60 years, without anybody reading this. So that was a revelation, and I immediately realized that whereas the nematode had extremely small cells, which we would never, we argued, be able to penetrate with electrodes, you could do the electrophysiology on Ascaris. So with that, and after some other things, we decided we would go forward with it. And the first thing and our plan was to reconstruct the entire nervous system. The whole organism has 1,000 cells. And so it looked as though maybe there were 300 in the brain. So that's something you can get what I call the wiring diagram. So we had to learn to do electron microscopy. We started genetics from zero, and we learned how to do it. And the idea was we will get mutants of behavior. That is, mutants in which the nematodes either don't move properly or don't move at all. And so a big project got started. Now, I should tell you that, of course, to begin with, there was nobody else in this except I had one technician. I taught her how to do all of the other things. We were making them, we had a kitchen that knew how to make media, so they switched over. The Petri dishes were almost the same. And what we did is we grew the nematodes on E. coli. So we had hardly a change. We just needed microscopes, a dissecting microscope, to be able to see the details of the worms. And after getting this going, we then started. I was very fortunate in that a friend of mine had given up his lab, and he had a very good technician that he wanted to give a job to. And so I gave him a job. He became our electron microscope a technician. He learned how to cut the thin sections, and he was superb. His name was Nickel Thompson. I then knew this would be a rather big project, and so we would probably need computers in order to help us with all the mapping and so on. So I took off time and learned how to program computers and to do things. And we began by having people come to Cambridge, and we didn't ask for any experience. Nobody had had any experience in this. They all learned. And one of them, John White, who ultimately landed up by helping with all the reconstructions. John White was a computer technician, computer scientist in London, and so I asked him if he would be interested. He was looking for a job, so we said, we'll hire you. Now, we did have a computer in the lab at the time. It was a computer that came out of a World War II submarine. It was a Ferranti computer. It had to be programmed directly in binary. You wrote the code words into it in binary. That was a very difficult thing to do. However, we got started on that, but realized we've got to get more sophisticated. And eventually, we were able to buy a kind of one of the first really modern computers. It was called a modular one. It had just a paper tape reader and about, I think the first one we got was about 128,000 locations. Of course, you know, nowadays it's all mega and tera memories and so on. But those were all hand-wired coils that we used as the switches. It was extremely slow. It was each program instruction took milliseconds to execute. Of course, they speeded up afterwards. So we got this, and there was a part of our building which had not been built on at all. It was just the building shell. We put down cardboard sheets on the floor, and we put our computer there. And we all worked downstairs doing a lot of things. Amongst the people who joined me was a chemist from Cambridge who had gone to the Salk Institute, which we knew, and he had worked with with Leslie Orgel on fundamental chemistry of how you make the nucleotides, things that if you'd come to the evolution meeting were discussed at the time of how all these biochemistry got established, with no one now ordering the chemicals, of course. And so he had come there, and it became clear that we could now do the total analysis of this organism. We could find out about where every cell came from, the lineage, and of course, then as time went on, more and more people joined us, many postdocs from America who were intrigued by the novelty of this. None of them had ever done anything in neuroscience or developmental biology, but they all came to learn. And so there's a long line of people who are all now pretty famous who came to work there. And of course, that work continued, and I won't give you the detailed steps because I think there's a take-home a few take-home lessons. See, no one had ever attempted to give a complete analysis of one organism, its genetics and its lineage, where the cells all came from in development. And what we knew and what we established was that this had a unique method of generating this nervous system, which was different in the two sexes. Different cells had different fates, so the versatility was great. And I think this now exists. You can go and look it up. You can tell now where every cell comes in development. And as many of you know later, the entire gene sequence is known. Many of the genes are characterized. And there is the possibility of trying to understand globally one organism, how it got specified and how it does this. And I think there are a number of things to learn about how you do this, which was that everybody who came to work on C. elegans worked on their own problems. We didn't have teams. Everybody did their own work, made collaborations with other people as were necessary, and a lot of the people who came are today, and of yesterday for some of them, the leaders in American and European science. And the criteria of making individuals responsible for the work is the only way to do this. So I am totally opposed to this business of now, which is developing, of managed science, of having big shot with several little shots underneath and then all the way down, working in these things. We never had lab meetings. If we did, the people were asked to come and they were asked to speak a half an hour before. So they said they can't do this, they need time to prepare. And we said, if you need time to prepare to talk about the problem you're supposed to be engaged in, forget it, you're not working here properly. So they all learned to speak about their work directly. And this business of giving responsibility, remember, all of these people had done PhDs in a wide range of subjects before and came into this quite uniquely and trained themselves to do this. So I think this is a very good way of having people work together, because each person knows he's key for that problem. If he doesn't do it, everybody else will fall down. But of course, everybody else is carrying on in this. The group was never really very big. I think at the height, we were 40 people in one group. But let me tell you, that group did lots of other things apart from the nematode. It also had a fly lab. It also had this, there was a wide range of work. And the old LMB was a very great place to work because somewhere in that building, it wasn't a large lab. It had it had a very small staff, had lots of postdocs and a few students. And I think that is one of the great ways, because you could always find someone in the lab who could help you with problems in mathematics. We had a terrific workshop. We used to build our own equipment. We built rotating anode tubes for X-ray diffraction. They're in the same building. So if you can think of a paradise to do science, that was the place. You could come there, you worked with someone to learn, and then you were on your own. And I think that is the thing. We also organized the services here. In the old days, you had to stand in line in front of the stores, hand in your request. You then had to sign, for 2 pipettes or a beaker or whatever you got. And we changed that. We had a kitchen like a supermarket. The front was shelves. You can see this design in many labs. The people at the back were told, keep the things full. And people were encouraged to just go and take what you need. They were not allowed to hoard anything. That we used to go and clean out their refrigerators to stop them from hoarding, because everybody could then get on with the science. And I think those were really, the 70s and 80s were really amazing years for the development of all of that. Now, of course, you say, what is the ideas behind doing what is now called connectomics? This is the name given to all of these branches. It was always omic science. And that's a mistake, because it isn't an ohm, because nomos is a message in Greek. So it's the genome. It's not the ohm of the gene. It's the gnome, which is the knowledge about it. However, they just had omic sciences. And as you know, if you go and read the modern literature, everything is omics. Okay? We used to say, how about home sweet omics? Your home is also an omic science. However, but I think you still have to justify to many people why neuroanatomy is important. Because if you look at nervous systems, they're not just plain networks. Everything is very carefully organized. The layers, everything is in layers. If you look at the cortex, the old cortex, the cortex that first there, which is the cortex that governs smell, that's got 3 layers of cell. The neocortex, including the visual cortex, is 6 layers. Right? So all of that developed, and the concept of how this nervous system expanded is tremendously important. But of course, you have to determine what connects with everything else, you have to show it's an invariant. And of course, the nature of the connection is called a synapse, as we know. And so for the C. elegans, we have the complete network. And many people are exploring this today in order to try to reconstruct what specifies it, and in order to find out how it actually works in a coordinated manner to control the behavior of this little creature that can smell things, can taste things, it has all of these senses, can't see, has no eyes. Okay, so because its habitation is in the soil where there's no light. So I think that was an interesting lesson in science. And of course, for us today, we would like to do this for human beings. We'd like to know, why is this nervous system that's in your head different from the one in your closest relative, the chimpanzee? Of course, it's well known there are no PhD students amongst chimpanzees, and chimpanzees are still wired to be adapted to their environment, to modify it, but they don't have this insight, this way of solving problems that is the particular human capacity. So I still think that is the most challenging biological problem that we still have to meet, by no means solved, by no means. And of course, the C. elegans was the forerunner of this, and many people now are trying to do the same with other organisms. Let me give you some example. It is surprising that they nearly all came out of the old molecular biology. So a colleague of mine from the old days, who's now, alas, no longer with us, Seymour Benzer, he decided about the same time that I switched to do the nematode, he decided he would switch as well. He decided to go to something which had genetics, which was Drosophila. So he left and went to Caltech and started to work on Drosophila. You see, and he used to say he spent his life working with E. coli, and if you like, E. coli is one cell, so it has one neuron itself. Okay, of course he'd like to work on the human brain, which is about 10 to the 10 neurons. It's a lot, 10 to the 10. So he said he chose the square root of that, which is 10 to the 5, which is about the size of the Drosophila brain. It's about 10 to the 5 neurons. And of course, C. elegans is the square root of Drosophila. It's about 300 neurons. So we've got these scales, and to go from the mouse is about 10 to the 9, or 10 to the 8, between 10 to the 8, 10 to the 9 neurons. So you've got a big jump to get up to the size of the primate brain. The rodents are the step just before the primates. The rodents are the primate nearest neighbors in the evolutionary tree. But, and if you like, the most original of the rodents is the rabbit, which used to be a great organism for immunology, to make antibodies in rabbits in labs. Of course, I think that to understand all of these pieces, we have to go back to theories of the nervous system and to theories of how they work. Because you'll find that many of the transmitters are found in C. elegans, which is very, very early in the evolution. It's an invertebrate, but it has all of the, it has acetylcholine, has glutamate, and you can find that these transmitters are all the chemistry and biochemistry is very old indeed. They're all derivatives of amino acids in some sense that act in the nervous system. Tryptophan, tyrosine, modifications of these form most of the transmitters. Some are excitatory and some are inhibitory. And they all work, as was shown, by controlling the influx of ions. And it's these ions that give you the so-called electrical properties of the neuron. And for a long time, these were measured with electrodes. So you could do this. There are many ways now to measure all of these. And so we think that if you had, at least that was the idea with C. elegans, that if you had the nervous system and you knew which were inhibitory, which were excitatory, you could work out all the steps in behavior. That still is not known in any detail, but it's beginning to come now. People have reconstructed exactly how C. elegans stops, changes direction. It's all guided by smell, chemotaxis in that sense, which acts through olfactory neurons, and which is the paradigm just exactly the way you smell things. C. elegans also smells things with old cilia. They used to be ciliated neurons. They still are ciliated neurons, but the cilia don't wave anymore. They're in your nose. They're your olfactory neurons. And you have lots of them, lots of different ones that can pick up various smells, and so has Drosophila. So there is a common unity to all of this, and we think that if we pursue this as we have been doing, then we can do, we will be able to put it all together. Now, it's not very easy to do this because still enormous things are just unknown. We just have no idea how the various layers are connected. We have a general idea. Part of the nervous system that we're beginning to master is the way the eye works, your eye works, you see. Because you have to ask yourself, how does the world stay still when you move your head? You see? If my eye was just like a radar camera, the world should move when I move my head. But I have an eye which can keep a fixed picture in front of me. And that's all automatic. I'm not preventing it. It just happens. Happens very quickly and in very great detail. So there's obviously, and that's governed by a set of nerves that come out of your brain. It's the ones that control the position of your eyes, how they move, and so on. So there is, even for this object, the eye, which is so important for us, there still is a lot to work out. There still are a lot of interneurons in the eye, where the various cells, we know you have eyes that see 3 colors, trichromatic, and you see them through two kinds of cones and one rod. The rod is particularly designed to be sensitive to single photons. It's a very sensitive object. And so this is how you see in the dark. And you need that retina in order to see, to use rods to see in the dark. The cones are less sensitive, but exquisitely sensitive to color. So that's how, when you look on the scene, you just take everything in. You take all the patterns. So that in itself is a big thing. Of course, if you put electrodes into the cones, you only see flashes. You don't know how the whole picture is integrated, because that's done by going through various stages till we get right to the back of the brain, where there's the visual cortex. And one of the people that came to work with us in the old days, and I'll tell you the story so you can see the connection to modern nuclear science, was a young man called David Marr. David was a student at Cambridge in mathematics. A rather very clever young man, and he went to work in London. He wanted to learn neuroscience. And when he left London, because we knew through other people, we knew him, we said, well, you can come and work in our lab. So he actually joined the LMB, never having done any biology except for his PhD, which was on the cerebellum. He had made a model of the cerebellum. Turned out to be wrong, just like most of them were at the time. But he came there. Now, when I was a student many years ago in South Africa, I had a friend who's about my age, who was a brilliant mathematician. His name was Seymour Papert. And Seymour came to work with us in South Africa when we were still just beginning to do research there. And then he wanted to learn the nervous system. So I said he could come. I would teach him neuroscience if he would teach me mathematics. He was an expert in Boolean algebra. Had discovered many things quite independently on his own. He then went abroad and finally landed up at MIT, working with a man who I got to know later in the artificial intelligence group at MIT. And I said to David Marr, I'll send you to Seymour. You go to MIT, you go work with those people. You can learn about that. You can learn about computers, we can't teach that to you. Because unfortunately in England, they had decided that artificial intelligence was not a field to enter. A big paper was written, and so they stopped supporting it. The people who were still doing it was a group in Edinburgh, a man called Donald Mickey, who I knew very well because he was married to Anne Mickey, who was a mouse geneticist and embryologist. And we had another friend in Cambridge who was the professor, one of the professors of theoretical chemistry, called Christopher Longwood-Higgins. He's expert. He was expert in statistical mechanics. And Christopher decided that this wasn't getting anywhere, so he decided to drop everything, resigned his chair, and he went with Donald Mickey. Now, Donald Mickey was an interesting young man because he worked with Turing during the war. Turing was employed by the code breakers. They broke a very important German code. And Turing was the man who generalized this. Mickey worked in this. The code was broken, but the Germans never found out because they had to show that they let things happen. They knew they were going to happen because had they tried to stop it, the Germans would have deduced this. So it was just random what they stopped. And so the Germans believed that they'd never broken these codes. They were and Turing was a great code breaker during the war. He built a machine to break codes as well. So Turing, you will remember, I mentioned him. He was a young fellow in Cambridge in the 30s. So Christopher went this, and he finally got supported by the Royal Society, but he became a professor. But someone had to be responsible to the Royal Society. And at the time, the secretary of the Royal Society was a scientist called Bernard Katz, who was a great neurophysiologist. And so he knew I knew both of them. So I got the job of going to Edinburgh and looking after Christopher there, which suited me fine because I was interested in the field of computational biology and computation. So if you like, there just were a whole lot of amateurs, basically, who were just jolly good amateurs and had no professional training in the field but got into it. Okay. And I think that is the criterion for doing really good science, not this narrower specialization until, as we used to say, you're the expert on the fifth base of transfer RNA for alanine. You only know about that one base, methylinosine. So I think there are many prescriptions for doing good science, and I think that throughout this period we learned about this, and we learned not how to organize it, we just let it organize itself. And in the end, if you look at these, Jerry Rubin came to the lab as a postdoc, as a student in Cambridge. When he came, I knew just leave him, he's good enough. And I remember he came to me, says, I think it's time I submitted my thesis. I said to him, what thesis? He says, my PhD thesis. I said, I thought you had a PhD. He was self-propelled. Today, he's the director of Janelia Farm Institute. This is one of the big new centers for neuroscience. He pioneered lots of things on Drosophila and eventually landed up there. So the good scientists don't worry about what degrees you have and all of this. They just worry about, can you solve that problem? How would you solve it? And I think in my life, I've been a member of all these institutions. I still am a member of Janelia Farm. I still get all their things. Of course, I can't go there anymore because my doctor doesn't let me travel. Even with Singapore Airways, I have to stay on a wheelchair. But still, that was a terrific experience of life, and I've always wanted to live the scientific life that way. Well, as you will hear on Monday when you come, there are more connections which we will explore. And one of them, Francis Crick decided after he tried developmental biology, and this is to get interested in the brain. So in 1975, he packed up and went to the Salk Institute in Southern California, La Jolla. It's a very nice place. And there he decided he would just do the brain. Now I'll tell you some funny stories about this. When he was in Cambridge, he had a big sign printed very big, about the size of the blackboard, and on it was written, "Reading rots the mind." You see? That is, he said, and I never knew where he got it from, but he had this. And of course, it was amusing because all he did was sit under the sign and read this. I later learned he stole it from Christopher Longwood Higgins's office, who really believed that reading rots the mind, and that you had to, thinking didn't, you see. And I remember going to see Francis in 1977. He had a terrific office, and every surface on it, there were about 7 tables there and a desk. Every surface was covered with journals, with copies of papers. But still above him, there was this big sign that said, "Reading rots the mind." So just be careful. You can read, but you must read and think at the same time. So Francis went there, and as many of you know, he became actually the head of the Salk at one stage, but since he got cancer, he retired from that. But he came with the idea that the problem to solve is consciousness. I disagree with this. I don't think consciousness is a problem that you can solve just as such. Because who has consciousness? Has the Elegans got consciousness? Consciousness means it's the real function of the brain. It's how the brain looks at itself. And I don't think there is a special circuit for it. He wrote a number of books. You can read them. I think there are a few good things in them, but I think let us put consciousness on one side for the moment and try to solve exactly how these circuits work, because there still is so much unknown. Right now, in many cases, people believe you needn't do all of this stuff on brains. You can just bypass it all and just do what's called artificial intelligence, which is you just need to do psychology. Just think of this as a black box which can sort of issue things. I tell you, the young man who founded this Google, which Google bought, the DeepMind company, his mother is Singaporean. He was, as a young man in England, he was an expert on Go, the game. You know, a genius, a prodigy. But in order to push what he'd been working out, he went back and did a PhD. He worked with people at University College who are very well known for their work on the hippocampus and on parts of the behavior of the brain. But he felt he needed the background in this machine. This is a machine. It's a machine you can control in order to pursue. And as you know, he solved a lot of problems, and it's today the hottest thing. Google bought his company, so he can now think about brains without having to look for bread somewhere. So I think that, and of course everybody thinks if we can get rid of people, that'll be the best thing. So we are now entering an epoch of driverless cars, driverless aeroplanes. And of course there are even some people who think we won't need scientists. We'll just do it. The scientists will be the machines. I used to be a great lover of science fiction stories, and I had a vast collection of the really great ones, the ones that have a deep message. And I'll just tell you about two of these, because they make you pause to think. All right? So, one day, in the story of this, these people land on Earth, and they've come there, and they bring a little black box which gives limitless energy. And they come there, and this reporter is very suspicious of them in the story. And he discovers they have a book that they all believe in. It's called How to Serve Man. That's the name of the book. And they're doing this. They then organize travel back to their home planet for people who go there, send messages back, we're having a great time, it's marvelous. The reporter finally discovers that by translating this strange language, that the book, How to Serve Man, is a cookery book. And that they are sending people back to eat them, you see. So it's just a joke, How to Serve Man. But there are these things that I think for you who are the next generation and who are all doing science, just try and look at this past, look at the individuals, see what they accomplished, because they are the people that you should follow. And you don't have to have big things, because you can be. I just give you one word of advice. If you're a rebel, you don't have to tell anybody you're a rebel. Just do it. Just do it. So I think it's all up to you, your personal responsibility. On Monday, you will hear from Terry Sinofsky. He's a professor at the Salk Institute, originally trained as a physicist with Wheeler, who's a very famous physicist. And Terry then went to study the nervous system. He switched as well. He's a very good mathematician, physicist, who now studies the nervous system. He's written a book on the history of all these attempts at artificial intelligence, and he will talk about this on Monday. It's a good book. It's in press now. If anybody wants to read it, I have a copy of it, which I'm happy to lend to people. All right. So I think you'll have had an insight in these 2 days now, and 2 days which will follow, over what I think has been a cataclysmic history of biology, biological science, starting in 1953. It's just a good point with a model of the double helix, right up to the new science which is now dominating the world. So please come on Monday, and I'd like to express my thanks to Balash and Jan here for helping to organize this, and also to NTU for its support of this endeavor. But I hope that when you're all grown up and are either generals in the army or managers of hotels or whatever it is you're going to do, that you will remember these formative years. Thank you.
More than anything, it's very motivational to come.
Can't hear, sorry.
More than anything, I guess it's very motivational to come and sit for the things. I feel that you should go on, and we should be here listening.
Well, you can come and listen on Monday, and Tuesday will be a free day for lots of discussion, because Terry is only going to give one lecture. So we thought he might give 2, but he'll be here, and we'll just have an open discussion on Tuesday.
I wanted to ask, what was the trigger for you to switch from the molecular biology to C. elegans? What was that one thought?
There's no trigger. There just is. It was just an interest. You see, one of the important things is when everything looked as though it was getting boring, you know, and that it was just turning the crank, then I get bored very quickly. In fact, often when I finish work, Francis Crick used to lock me in a room and never let me out till I wrote the paper. I just feel the paper, it's boring. I know the answer, it's enough for me. So you have to be a bit restless, you see, and you have to say, oh, well, who wants to turn the crank, you know, another sequence. Doesn't tell you much more. So once it gets boring, that's the challenge. Then you must go to another. And I think that's the history of science. Science, it's the pursuit of knowledge. Knowledge is tremendously important. It's what distinguishes us from the chimpanzees. Okay, because that's why I think there's no trigger. You don't wake up one morning and say, I have seen the light. I have only seen the light when the light is shone on it. You see, as I did when I saw that model. The model was just the moment of transition. It was a jump. But we were all trying to solve the problem, you know, in some way or another. So I think it's very hard, you see, and that is why the double helix has become such a symbol of the time. Everybody has it in their logo. Yes. In fact, I made a logo for someone, which was a medical thing. And as you know, the sign of medicine is the rod of Aesculapius with the two snakes. It is believed that he used that rod, to draw roundworm parasites out of people. They're not snakes. They are, in fact, nematodes. And that he used them to pull the nematode, the tapeworms out. They were. But that doesn't. So, but if you wind the snakes as a double helix around the rod, then you've got the best logo of all. Okay.
Thank you.
Thank you very much. That was very intriguing talk. I'm Madhura, a postdoctoral fellow from LKC School of Medicine. I'm Madhura, a postdoc fellow from LKC School of Medicine. And I have a question to ask as a young scientist for us to follow today. So you mentioned many important points in your talk about how it is important to pursue a good question and not worry about degree and stuff. And I appreciate that you took many people on board and trusted their intelligence, and just based on the trust, you took them on board, let them do what they wanted to do. But in today's world, as a young scientist, it's pretty hard to do it. I mean, the way we see the world today as a scientific world. So if we do have a question to pursue in reality, how should we navigate today's world so that we get to work on it? Because,
I think, of course, you have to start by learning one technique. So just learn a technique, and then you can use that technique to go. That's why I think experiments are so important, that you learn from an experiment what it is, how to intervene in nature. And I'm still an experimentalist, even though I find it a little bit difficult to do experiments now. But I think that's the key thing, is to have something like this, which is a discernible target to get. So I think it can be done. If those of you who know my colleagues here in MEL, which is called the Molecular Engineering Lab, I must tell you how I got to that. They are all, some came to work with us when they were still students, and they just got into the group and they worked on a project which we did here in Singapore with a farmer's father, Venki, and it was to sequence the pufferfish. Now, why the pufferfish, you ask? What a strange, strange business to go and look. It's because I discovered, reading in a library in Woods Hole one summer, I discovered that pufferfish had very small genomes, about 1/10 of the size of this. So I immediately said, if we want a higher organism genome, that is the first one we should sequence. Because, and of course, we spent a lot of time using the pufferfish genes. So a lot of people who'd never done anything on genes came to work with us, and we'd say, well, why don't you get this gene out of the pufferfish and characterize it as your project? And we did a lot of that over the years. And when it came to sequence this, they wouldn't support this because it was now already an international project. And fortunately, Singapore gave me some money to support it. But we didn't have the machinery here or anything. So I phoned up Craig Venter, someone I knew. And we knew he had lots of machines, could do it. And I said, Craig, I've got a project for you. Oh, he said, that's good. What is it? I said, its first object is to deal with Eric Lander, who was, of course, this. So he said to me, I'm in. I said, look, we want to sequence the pufferfish. It's 1/8 the size of the human genome. It's 400 megabases. We can do it. And he said, we'll do it. So fortunately, Singapore gave me money, and I was able to pay Venter. But I said, it's not your project. You can have one of your people on the paper, but only one. It's our project. So we sequenced, just by using that whole institutional apparatus that he put up there at the time. And of course, it's turned out to have tiny genes so that you could get pieces that you could clone in lambda, which had 3 genes, 3 higher organism genes. They were tiny, and in this way, we managed to characterize lots and lots of the machinery of higher organisms, and quite often use those sequences from the fish to probe for the human sequences. But the whole genome could be found on one library of 20 kb segments cloned in lambda. We made these libraries, and we just had this. So anybody who came to the lab and wanted to do something, we say, well, why don't you try and get the insulin gene out of this, or whatever. And then many people actually started their whole career in genetics by working on the fugu, the pufferfish. So it was, again, we used to call it the discount genome, because it gave you 80%. You see, being 1/8, it was, whatever it is, it was about 90% discount on the sequencing. You could just do it. So a lucky thing like that. And that was, I discovered, in a journal which hardly anybody read, but I found this. And I thought, this is marvelous. No one else knows about this. So let's go for it. Of course, you see, I then learned that fugu is a delicacy in labs. I then learned about tetrodotoxin. Okay, and we've solved many of the problems about that. And I know that people used to take me in Japan to very expensive restaurants to eat fugu, which is a great delicacy. And they used to say when I ate it, can you feel the tingle? And I used to tell them, look, if I felt the tingle, it would mean I'm dead. So whether I tell you, it doesn't matter. But today, most of the delicacy, most of the fugu is cultivated, so it doesn't get infected. The fugu gets infected with this bacterium by eating clams, which are infected. And so the bacterium hides away in the fugu and makes tetrodotoxin. But the tetrodotoxin is only made after the fugu dies, you see, because you realize for a bacterium there is a problem if you live in the ocean. What happens when your host dies? How do you get another host? So many bacteria had become luminescent. They shine in the dark. So other fish come and have a look to see what it is, and meantime the bacterium can hop on another boat, because you realize the ocean is just, animals are little islands in this ocean. So everybody adapts in that way and finds some way to continue their existence. So it's well known, all of this, how in fact we know what switches on the synthesis. We know what switches on luminescence. It's arginine. Free arginine is the inducer of this chemiluminescence. So you learn a lot just by going to restaurants.
May I ask one more question? I'll take you back to C. elegans. So when you read about Ascaris and other micrometazoa and then decided to work on C. elegans, so was there anything known about C. elegans at that time?
DNA of?
Was there anything known about C. elegans at that time when you decided to?
About DNA? I didn't hear.
Was anything known about C. elegans by the time that you started working on it?
Oh yes, it was known just as an organism. There was a little bit of work. Someone had tried, a man called Doherty, had tried to cultivate it on a synthetic medium. So there was a little bit. And a man called Nigon, who actually is the discoverer of C. elegans, the first isolator, was in Paris. But he was just a classical nematologist who just cultivated it. It was known that there were males and females, but very little was known other than that.
Thank you.
So we had to start pretty close to the beginning.
A lot of time. Anybody? Sure.
Sydney, I had the providential accident when I was a young PhD student to meet Donald McKee, whom you referred to.
Donald McKee?
Yes.
Yes.
He was the professor of informatics and neuroscience or something
yes.
like that. And he was a fundamentalist Christian. I don't know which denomination he belonged to, with 5 children and a big family and so on. But he believed in downward causation. And he wrote a paper, an information theory paper, downward causation in the brain.
Yeah.
So what do you think about the concept? It's a very controversial concept.
I don't know that paper very well, because he was in Edinburgh, you see, and I must say I haven't read much. I only know Mickey's history of his connection with Turing, because you see, when after Turing committed suicide, and it was a great shame, but his mother never believed it, and she saved everything. And when she died, she left all of his belongings, and there was all his money was left to something called the Turing Trust. And I was a trustee of that. Turing Trust did a lot of things. it enabled a life of Turing to be written. It's quite a famous book. And it also gave out a few scholarships and things like this. And I met Donald Mickey and a few others who are now dead now as well, who were early collaborators of Turing. So I didn't know much about Donald. I knew his wife better. She was a mouse embryologist. And they both got killed in a motor car accident. You see, so that's the thing.
All right. Next one. Yes.
So you were saying earlier that you allowed your postdocs and students to kind of a bit start something they were interested in, although they had to be a part of the whole lab. So I guess that process would take time. How were you managing the fact that I guess you needed to justify fundings and things like that?
Well, my students allow me to be part of the lab now, today. No, I think, you see, I believe that if you're given the responsibility, you will accept it and do it. And I think that has always been the way we've run science from the early days in Cambridge. Long ago, we've always said we didn't like this business of having, a kind of hierarchy of managers and sub-managers and sub-things. I also never liked what are called group meetings, because there's always one guy who sits in a corner and doesn't say anything. There's always another one with a big mouth who keeps on talking all the time. It's often the people who sit in the corner and don't say anything are the only people who are worth listening to. So I think that these things which just are not creative in any way, they just regurgitate what has already been digested. It's kind of, and then they do the who's responsible for this part of the lab and so on. So I don't think I like to run things that way. In fact, I don't like to run things. But basically, the thing is, I think we've had, you can speak to the people who are in Mel. We've had people have left, they've gone to good jobs elsewhere in Singapore. And of course, if they don't take the responsibility, then they leave and don't do research anymore. So it's good. If you keep people from wasting research money, it's also good. They become managers. It's fine. I don't say that's an inferior thing. It's not something. I don't know how to teach management. I think management and cooperation is something you've got to learn. When I say a team, I don't mean a team with a leader and two deputy leaders and then two deputy leaders. I don't mean a hierarchy. Because today, most American scientists, I remember there was one, this is a big joke used to be told. He ran a big group in University of California in San Francisco. I think there were 65 people in his group. and they used to have a seminar. They used to have a visiting speaker. And at the end of the talk, he made a move to ask. He said he didn't believe what the speaker said. So he made a move to ask him a question. But his assistant was there. She pulled his arm and she said, "Be careful. You actually wrote a paper on this subject last week." He didn't know. He didn't know. I knew somebody had 3,000 papers. He was the head of the Sloan Kettering Institute. He'd just forgotten what he was an author of, because the papers were written by someone else and his name was put at the end. It's what we call the caboose theory of writing papers. That's, you see, you put the senior author at the end. So, you see, and the people in the middle are all anonymous. They might as well be called number 1, 2, 3, 4, 5. Forget it, you see. It's the first author and the last author. On these papers, we're often, I don't know, I think you should have a look at some of the papers coming out of CERN, what's atomic energy. The list of authors and their addresses is longer than the content of the paper. This is a list which is about 3 pages, and then the paper is about 4 lines. And that's what it is. Because, of course, those are gigantic engineering projects. Anyway, I don't envy you, you know, because science has become so structured these days, and you've got to have so many permits to do everything. So, but I think if you can find congenial people to work with, that's the important thing. I'm so pleased that looks like the average age here, even though of Balazs,
Details.
is well below 40, well below 40. That's good, because you are the future, you see. Don't ever forget this.
That's to me.
Come on Monday, that's
Right,
still in the
exactly.
future, please.
This sounds like an excellent moment to stop this session. Monday is important, that, as Sydney said, to come. Sydney will be here as well, but Terry Sinofsky will be here, and the rest of the way we run it will be the same as we have it now.
Yeah.
So it's all very much without surprises. But it's going to be very exciting again. So thanks. Let's thank Sydney once again. And I wish you all a very good weekend after you had coffee. And